Thank you for the introduction, and thank you very much to Karen Reed and Francis Riekoff for this invitation to present here at FrieslandCampina Institute. And the topic I'm going to talk about here is protein from meat, dairy or plant sources. What's the difference for aging muscles? In terms of the structure of this presentation, I've split the presentation into 4 primary sections. The first section is to highlight the importance of skeletal muscle mass for healthy aging. The second topic is to establish the key factors that determine the anabolic potential of a protein source, and we'll focus on 3 primary factors. I'm then going to spend most of the time comparing the anabolic capacity, which we're going to measure in based of potency in terms of stimulating muscle protein synthesis of different animal versus plant-based protein sources consumed in either food or supplement form for older adults. And then finally, we're going to talk about this idea of protein quality and where next for scientific research. So by way of introduction, I'm sure it's not of any surprise that with advancing age, there is an inevitable loss of skeletal muscle mass. And of course, the trajectory of this muscle loss is dependent upon several factors. And one of those factors is muscle disuse, which can consist of acute periods of disuse, i.e. a short-term period, say on crutches, for example, and range into more extreme periods of muscle disuse, such as leading to a bed rest scenario. And these periods of disuse leads to a greater trajectory of muscle loss with advancing age. But what's important from a healthy aging perspective is trying to maintain that muscle mass above this threshold of disability. If we can achieve this and maintain sufficient muscle mass above this threshold, we're able to live healthier lives for a longer time period, i.e. maintain that independence into later life. Before we go into nutritional interventions to offset the loss of muscle mass with advancing age, it's important just to review the different series of factors which contribute to muscle loss with advancing age. So here we have the muscle of a 25-year-old, which we can see the bone in the middle, muscle just on the outside, and then on the outside again is fat mass, compared to an older adult whereby we've got this infiltration of fat into the muscle tissue, and of course showing less muscle compared to the younger muscle tissue. But what you see in this diagram is a series of different factors, which in the red include these internal factors that cause muscle loss. So for example, low-grade inflammation and the hormonal changes we see with advancing age. We've got these external factors, and inactivity, injury and illness and insulin resistance are several factors. But we've also got this concept of anabolic resistance, which essentially refers to the reduced sensitivity of skeletal muscle mass to exercise and or nutrition. And it's this reduced sensitivity of muscle protein synthesis to these anabolic stimuli which describe anabolic resistance. So when we talk about muscle protein synthesis and muscle protein breakdown, essentially we're talking about these 2 processes which are observed on this diagram here. So when we talk about protein synthesis, we're referring to the synthesis or the incorporation of amino acids either from the blood or the intracellular pool of the muscle into bound muscle proteins. And these muscle proteins can be crudely classified either as myofibrillar proteins, which include actin, myosin, tropomyosin and troponin, so these contractile proteins, and also mitochondrial proteins. So these are the energy producing proteins. And we also have another metabolic process, which is called muscle protein breakdown, which refers to the breakdown of these muscle proteins back into their amino acid precursors. And essentially it's the balance between muscle protein synthesis and muscle protein breakdown, which determines whether there's a gain of muscle mass or a loss of muscle mass. So if muscle protein breakdown exceeds protein synthesis, we get this loss of muscle protein or muscle protein mass. Whereas if muscle protein synthesis exceeds breakdown, we get a gain of muscle mass. But out of both of these 2 processes, it appears that a reduction in muscle protein synthesis with aging is primarily responsible for the decline in muscle mass with advancing age. So if we can implement nutritional strategies to attenuate this decline in muscle protein or decline in the impairment in muscle protein synthesis in response to nutrition and or exercise, then in theory we can combat this loss of muscle mass with advancing age. Now this slide provides a classic experiment that demonstrated experimentally this phenomenon of muscle anabolic resistance. So on the y-axis we've got muscle protein synthesis rates, and on the x-axis we've got different doses of essential amino acids. So here we've got the basal condition, so 0 grams of essential amino acids. Here we've got low doses of essential amino acids, and then here we've got high doses, so 20 grams of essential amino acids being equivalent of around 40 grams of protein. In the blue we've got the young, in the red we've got the elderly. And what you see is that at these moderate and even high doses of protein, you get this resistance or reduced response of muscle protein synthesis in the elderly compared to the young. Whereas in the basal condition, there's no differences between the age groups. So from a protein nutrition perspective, there's been a lot of interest in trying to determine the most beneficial protein sources for maximally stimulating muscle protein synthesis rates. And essentially there are 3 key factors that determine the potential of a protein source to stimulate muscle protein synthesis. First of all, one factor relates to the digestion and absorption kinetics of that protein source. The second factor relates to the essential amino acid profile and content of that protein source. And then finally, the third factor relates to the leucine content of that protein source. And we're going to go through in the next few slides differences between different protein sources in terms of these 3 different factors, and then look at how these factors impact the anabolic response to different protein sources. But before we do so, an analogy that myself and others like to use in this context to really highlight the role of, or the importance of, a full essential amino acid profile and a high leucine content for stimulating muscle protein synthesis is this brick wall analogy. So when we think about this brick wall analogy, we think about Lego, these Lego bricks that make up the brick wall, which we're going to that are going to represent muscle protein. At any one time, there's an endogenous amino acid pool which contains both the non-essential amino acids and the essential amino acids. But if we think about this brick wall analogy as, say, a building site, to be able to build this brick wall, we need a constant supply of protein, including a form or a supply of the essential amino acids. So the brick wall is made up of a number of different colored bricks which represent the amino acids. Here we've got the essential amino acids, which include the different colors of bricks here. You may well have heard about the mTOR pathway. Now, this pathway is a cascade of proteins that are found inside the muscle cell itself, which need to be switched on before this process of muscle protein synthesis can be triggered. And the red brick, the leucine, refers to a cup of coffee, which essentially activates this pathway to trigger this process of muscle protein synthesis. So what you can see here is that to stimulate this process, we need sufficient leucine, but we also need a complete profile of all these essential amino acids to build a complete brick wall and therefore stimulate muscle protein synthesis. So let's have a look at the essential amino acid profile and leucine content of a variety of common animal and plant-based protein sources. So what I've got on this diagram is, in the white, we've got the dairy proteins, and the red, we've got the other animal proteins, and then we've got green, we've got the plant-based proteins, and then I've added, just for good measure, the human muscle itself. So this is, if we were to take a muscle biopsy, mash up that biopsy, look at the amino acid content of that biopsy, here we've got the essential amino acid content of that human muscle itself. And here on the y-axis, first of all, we've got the essential amino acid content expressed as a percentage of that total protein content. And what you see here is that without exception, the animal-based protein sources, and in particular the milk proteins, have a higher essential amino acid content compared to the plant-based protein sources. And it appears that the milk proteins are the ones that contain a very similar amino acid or essential amino acid profile compared to human muscle itself. What's also interesting and denoted by the black font is that certain proteins are deficient in at least one essential amino acid, at least in comparison to FAO guidelines. So, and it appears that these are from the plant-based proteins. So here we've got the amino acids that these different plant-based proteins are deficient in, which generally speaking are either lysine or methionine. But what is interesting is that they're not necessarily deficient in both, and that has implications when we make recommendations further down the line. If we look at the leucine content of plant and animal-based protein sources, again, we see a similar pattern where, generally speaking, the leucine content of the dairy proteins, in particular whey protein, is relatively high compared to most of the plant-based proteins, although there are some exceptions in this regard. So that previous slide really walks through the differences between commonly consumed protein sources in terms of essential amino acid content and leucine content. But of course, what's most important from a scientific perspective is really evaluating these studies that have directly compared plant and animal-based protein sources in terms of their capacity to stimulate muscle protein synthesis. And to my knowledge, this was the first study that made that direct comparison, whereby muscle protein synthesis rates were measured in response to either the ingestion of skimmed milk or a soy milk condition, matched for dose. It was 18 grams of both conditions, ingested immediately after an acute bout of resistance exercise. And this was conducted in trained young men. So I appreciate not necessarily applicable to the older adults, which is the theme of this discussion, but this was the first study really to directly compare protein synthesis rates between the two different sources. What they showed was a greater, over 30% greater stimulation of muscle protein synthesis with skimmed milk compared to the soy milk condition during that acute exercise recovery period. And the three factors that potentially explained this greater response related to the greater digestibility of the skim milk, the more favorable essential amino acid profile in terms of a complete profile, and also the higher leucine content of the skim milk compared to the soy milk. Now, of course, the next logical step in terms of identifying the differences in anabolic capacity between protein sources were to take this milk protein condition and try and determine which fraction of that milk protein was particularly responsible for stimulating the greater response of muscle protein synthesis. So again, we've got muscle protein synthesis on the y-axis, and this time we've got on the x-axis, we've got a rested condition and an exercise condition in the red, solid red with whey, and in the hatched with casein. And of course, the dairy proteins consist of 80% casein and 20% whey. So this was a single leg exercise model whereby one leg exercised and the other leg remained rested to enable an internal comparison of both resting and exercise conditions within that same participant. So a really powerful study design. And what they showed in this study, and again, this is from Stuart Phillips' lab, Nick Bird was the first author. Actually, this was Luc van Loon's lab, this study, showed that whey protein stimulated a greater response both under resting and post-exercise conditions compared to the casein condition. Other studies have also compared between whey protein and plant-based protein sources. So this was a study again in older adults. And I should have said this study by Bird et al. Was also in older adults, where This time it was a dose response comparison. So whey protein was compared with soy protein at 3 different doses, 0, 20 and 40 grams of protein. And this time it was in post-exercise conditions. And what they showed here is that at each of the different conditions, there was a greater response of muscle protein synthesis with whey protein compared to the soy protein. Not only have milk proteins been compared to soy, but also beef has been compared to soy in terms of capacity to stimulate muscle protein synthesis rates. This was a study in middle-aged adults. These aren't actually published, but they are buried in a review paper, but Stuart Phillips gave permission to present these. What we have here is, again, resting versus post-exercise conditions. And again, we see a greater response of muscle protein synthesis with the beef compared to the soy. A final study, again in older adults this time, compared casein protein with a wheat hydrolysate, and they also had a hydrolysate casein condition, but the data aren't presented on this figure. And similarly shows that the response of muscle protein synthesis was greater with the casein condition compared to a dose-matched wheat conditions. This was a higher dose, around 35 grams of protein in both conditions. And again, they attributed the greater response with the casein to a favorable essential amino acid profile and a favorable leucine content. So when you take these several studies together that have directly compared between animal and plant-based protein sources, the general consensus based on these data are that animal-based proteins are more potent in terms of stimulating muscle protein synthesis in older adults compared to plant-based protein sources. However, of course, this statement is very much limited to the comparisons that have been made, given that we only really have comparisons to, at least when we're looking at plant-based protein sources, to wheat or to soy protein. And of course, there are several other plant-based protein sources that are ripe for research in this area. And there are a couple of labs that are very much looking at these all other plant-based protein sources alongside other alternative protein sources. And of course, this is an important notion given when we talk about protein recommendations. Not only do we need to be thinking about muscle protein synthesis and muscle metabolism, but we also need to be thinking beyond the health outcome markers and also think about sustainability as well. And of course, we know that from a environmental perspective, and this is very much a crude diagram of the greenhouse gas emissions that are associated with the full life cycle, processing life cycle of different plant and animal-based protein sources. Plant-based proteins have a lower carbon footprint compared to many animal-based protein sources. So of course, this is an area that we need to be well aware of. So moving forward, we need to think about other plant-based protein sources as well and work out what kind of recommendations we can make if we are going to perhaps increase the intake of plant-based protein sources. And of course, the most logical first step is to think about what recommendations can we make in terms of tailoring the dose of protein. Now, this was a really nice figure that was really put together from Stefan van Vliet in 2015, which was a hypothetical diagram based on this idea of the leucine threshold, i.e. how much leucine in theory you needed to maximally stimulate muscle protein synthesis rates in a given protein-rich food source. And if we take around 3 grams of leucine being the optimal level of leucine intake to maximally stimulate protein synthesis rates. So what they did here was just in theory, try and model how much of that protein source would be necessary to be ingested to stimulate muscle protein synthesis rates. And that's what you have in the black font here. So for whey protein, around 27 grams in theory of that whey protein would stimulate a maximal response in older adults. 875 mils of milk is what they came up with. So large glass or larger glass and a half of milk and so on. But what you see at the other end of the scale is how much of different plant-based protein sources you would need. And if we take potato protein, based on their modeling, it came out around 2,891 grams of potato protein, which is a lot of potato, i.e. around 13 large potatoes, which even for large eaters is pretty impractical. So another strategy may be in terms of increasing the anabolic potency of plant-based protein sources is to fortify these plant-based protein sources with additional leucine. So one example is shown here, albeit using an animal model, whereby whey was compared to wheat protein, the same dose in this scenario, and then also had a third arm that fortified the wheat protein with additional leucine, so that the leucine content of the wheat now matched the leucine content of the whey protein. Muscle protein synthesis rates were measured, albeit in a rodent model. But what you saw here from these data is, as you'd expect, a decreased response of protein synthesis when wheat and whey were compared on a gram for gram basis. But then when enriched with the additional leucine, there was a similar response of muscle protein synthesis. So suggesting that fortifying plant-based protein sources may be an efficacious, maybe a effective way to increase the anabolic response. The other way to think about how we can increase the anabolic response to plant-based protein sources is to really identify those plant-based protein sources with favorable essential amino acid content or leucine content. And there's a couple of examples here. But perhaps the best way is to think about complementary plant-based protein sources. So what we have here is the leucine on the left figure, methionine on the right figure. We've got the dotted line is a idea of where the sufficient level of that essential amino acid is based on FAO guidelines. And what you generally see, as mentioned earlier, is if we take pea protein, for example, it has a sufficient lysine content, but it's deficient in methionine. Whereas if we then take corn, for example, it's sufficient in methionine, but is deficient in lysine. So essentially, these are complementary plant-based protein sources which would cover all essential amino acids if paired together. And the same applies to when we combine soy protein with hemp, for example, potato protein with brown rice. And of course, there are a number of different ways we can do this, from recipes and different meal plans that could be put in place to ensure that these essential amino acid profiles are covered within a meal. Just to finish up, really, in terms of other areas of interest, there's been a lot of interest in alternative sources of protein, and insect protein is receiving much more attention in recent years, not only in other parts of the world, but also in Europe. Here we've got just very hypothetical, but the amino acid profile of, and in particular, the leucine content of various different insect protein sources. And as you can see, they're fairly favorable in relation to other commonly consumed food forms as well. So here we've got termites, grasshoppers and locusts, and crickets, and also the beetles showing quite favorable leucine profiles. And I think in the future will become much more acceptable as a protein source. Again, moving forward, I think there's going to be a real push to not only look at protein recommendations for health and well-being, but also marry that up with sustainability and environmental impact. And I think a good way moving forward is this mathematical modeling approach, whereby we can look at small tweaks in diet, perhaps changing slightly the meat content of the diet, for example, reducing the meat content and replacing that meat with some plant-based protein sources, and the impact that could have on both health outcomes and also environmental outcomes. So very much a balancing act, and I think mathematical modeling could be really effective in this regard. So the take-home message is, hopefully I've convinced you that The age-related decline in muscle mass is mediated in part by muscle anabolic resistance, i.e. this reduced sensitivity of muscle protein synthesis to ingested protein. There are three key factors which determine the anabolic potential of a protein source, namely the digestibility and the absorption kinetics of that protein source, the essential amino acid profile, and also the leucine content. On a gram for gram basis, dairy and meat proteins are more potent than soy and wheat for stimulating muscle protein synthesis rates. But of course, this comparison is limited to only soy and wheat protein. We don't know that much about some other alternative plant-based protein sources. And as mentioned, future protein recommendations should take this more holistic approach by considering not only health outcomes, but sustainability, appetite, and other nutrients of concern. And I think mathematical modeling is a really powerful approach to achieve that. I'd like to finish by thanking all of the people that helped me put this presentation together, from collaborators and colleagues. And also thanks again to FrieslandCampina for this opportunity to present here at this symposium. And thank you very much for listening.